7:20 to complete the exam. The second announcement is for homework questions. I'm going to ask you to come to office hours tomorrow, 10 to 12, or to email me your questions. And that's because I know some of you have questions about homework questions, especially the ones that I posted. Hopefully not, but if you do, well, I'm going to sort of, recap some of that as well. But I want this review session to benefit to everybody. That's why I want to make it like we'll go back to the slides and see if there are problems here and there. Because if we start like focusing on one problem or the other, many may not actually benefit from this. That's why. So let's get started with the homogenetics. We took 2 full lectures on that. So we're going to go a lot faster. And this is not going to be where I'm going to explain everything, but I'm going to stop, many points, and I'll sort of tell you the big lines of this, and also tell you in some cases, this is not something that you're going to be responsible for. So the first big line here was essentially that was the framework for the whole chapter, this whole set of lectures in development. You have different sets of activities and processes. You have positional information with the flag model. You have induction of notably several cell and single cell. And you have inhibitory activities. So the first one, the positional information, that's the model of the French flag, which I described to you. And the key point here is the influence of morphogens. So this definition of morphogen is very important to remember. And bacoid is a morphogen. It works. And I told you what it does, so it's easy to say now. Bacoid exerts its effect on transcription regulation gene expression through its concentration. You might remember that we talked about vulval development in C. elegans, and we talked about Lin3. This diffusible molecule, that's not a morphogen. Lin3 is not a morphogen. Okay, it doesn't work relative to its concentration. The other 2 activities, we have induction and we have inhibition. For this, we actually took one single example, which is the vulval development in C. elegans. And I just talked about the P here. This is the Lin3 LED23 pathway. I'm going to show you again the slide. And in the same system, we have an example of inhibitory activities that we see. That is the Lin12 pathway. LAC2, Lin12. LAC2 being the ligand and Lin12 being the receptor. We're going to go through this again. But this is just the structure of this chapter, okay? And think early fly development, bacoid as an example of positional information. Vulval development, we essentially go to see both induction and inhibitory process. So this is important to remember, this set of drawings in the middle, and I think I insisted on this notion of syncytium, and the general architecture of the larva and of the adult fly, and it's a 8-3-3 plan, 8 abdominal segments, 3 thoracic segments, and 3 head segments. So for the fly part, which occupies more than half of this lecture, this is really the crucial point. I think you spend 80% of your time reviewing those slides on this slide, and you'll be fine because it has pretty much everything. So it has this notion of gene hierarchy, the coordinate genes following gap genes, parallel genes, segment polarity genes, and I added to that homeotic genes. So remember, they are part of the hierarchy and they're all the way at the end. I'm not going to go back to what these genes do. Now, let's start questions. You have an example for each of these genes. I would retain the ones that I have circled. Bicoid, hunchback, even skipped, and hedgehog. The rest, don't even worry about it, but I want one of each. I'll tell you about homeotic genes later on. And in which category these genes fall. So even skipped, I sort of spend some time on even skipped, especially on the regulatory region, that's a parallel gene. A hedgehog, I spend some time on hedgehog. That's why I circled it. And it's a segment polarity gene. So that slide, I think, is a bit surprising. It's important to remember for maternal effect genes that really that has nothing to do with contradicting Mendel in any way. It's just a particularity of the early development in the fly, and that is the female that produces the oocytes actually produces RNAs, mRNAs that are transported into the oocyte. And this leads to the notion of maternal effect genes. And the genetics is that everything depends on the genotype of the female, of the mother, if you will. So if the mother is mutant, all of the progeny, no matter what is the male in this case, the father, all the progeny, even if it's zygotically wild type, homozygous, is mutant. And that female is the product of a cross between two heterozygous parents, one heterozygous female and one heterozygous male. Alternatively, and that's the other way, because otherwise you cannot generate an adult female that could lay eggs. If this female had its mother, so to speak, be homozygous, that wouldn't work, because that fly would never develop. So you had a homework on that. That's important to know, like, to understand, this table and this notion of maternal effect. So then I explained, I'm going to go skip to that. Talked about bicoid, and I explained production of gradients in the oocyte from the anterior to posterior. So these graphs are very important. So people ask me good questions about these graphs, and for good reasons, because this is a key point, but it's also something. There are things that are easy to overlook. The first thing is what the axes are. So the x-axis is position in the embryo, and the y-axis is amounts, basically. And see, I underlined that the top one represents amounts for the messenger RNAs that are labeled here, and the bottom ones for the protein, not the same thing. So you have to be careful to understand the distinction between the two, and you see that notably for hunchback, which is a gap gene, under the control of ubiquitin nanos, you see that hunchback mRNA is stable throughout the embryo, but you see the protein follows completely different pattern from the mRNA. So you have to understand that. And so then we move on to, from the morphogens, nanos, and bicoid, we move on to more complex expression patterns that are shown in the gap genes. So are there any questions? Something you'd like me to elaborate on? No? Yeah.
Yeah. The book says that the Eve gene is expressed in stripe. I think it is due to the certain combination of those proteins that are present in stripe too. And does Eve work in say stripe 4 under a different set of combination of different proteins, or does it just work on stripe 2?
So the answer to this question is that even is expressed in 7 different stripes, stripe 2 and other stripes as well. So in answer to your question, so that's the answer, right? Now here, look at this panel. So let me describe this for a second, because that's going to answer your question. You see these 7 stripes here, 1, 2, 3, okay? One of them has a different color. But you see 7 stripes. This is the pattern of expression of Eve. Okay? The blue, or bluish, this comes from the LAC-Z gene, beta-galactosidase, that was put under the control of this little piece here. So in the embryo, resoundingly, the answer to your question is, it is not only expressed in stripe 2, it's expressed in 7 different stripes. It's expressed in stripe. This is the 3rd stripe, 4th stripe, 7th, okay? But the key point is that the expression pattern responds to essentially different activities. And you sort of said that in your question. That was absolutely correct. But this is actually a fascinating point that they don't go through that much, is that why is it that in stripe 2 it is this little element here that's on, if you will, that controls the expression and responds to these factors, bicoid and hunchback, and these other elements are not influencing the expression of Eve. So that has to do with chromatin structure. Did I answer the question? Great. That was an excellent question. It shows that you understood that. Okay. Okay, so following that, so it's good because in fact in answering that question, you know, we talked. I just essentially summarized this slide. The complexity in the regulatory region of pair rule genes that are downstream from gap genes in the hierarchy. And this is even a simplified view. There are only 4 factors that are actually indicated here: croup, bicoid, hunchback, and giant. Actually, even more complex than that. And the regulatory region controlling the transcription of the gene is essentially an integrator of the factors that are present in each segment. And transcription being the readout. The readout sometimes is on, and sometimes is off. And then back on, because that depends on other things, can depend on giant, can depend on group or whatever. Okay. Now, See, I talked about fairly generally about this. This is what I want you to retain. Okay, I'm not going to put in like the Eve strike 2 promoter and ask you, what regulate the expression there. That would be a little too much. Okay. So now the homeotic genes. And then we remember, we're going to talk about segment polarity a little bit later, because I wanted to sort of, talk about hedgehog a little bit more. So now we're going to, skip over segment polarity, but they're downstream of the hierarchy. Now homeotic genes, and these are fascinating genes that are present as clusters. So in flies, there are 2 clusters, and you can see in evolution that they are part of groups of genes that are also essential for mammals. So since I know this is a review session, I'm not here to be all poetic about this, but without fly genetics, we wouldn't know anything about mammalian development. It's because of these guys who had isolated these mutations and cloned the genes that we had a genetic handle on Hox genes in the mouse, and we understood how the limbs are formed, how the rib cage is formed, how the neural tube, in case of hedgehog, is formed, etc. I'm losing time. I'm going to stop saying this. So anyway, what you have to retain here is thorax complex and Tinnipedia complex. And Tinnipedia being essentially an interior, involved in anterior segment identity, and bithorax, abdominal segment identity. I want you to remember UBX. And for the Tinnipedia complex and Tinnipedia, there are 5 genes here, but remember, Tinnipedia, exterior identity complex, and UBX is important for segment identity of the abdominal region. And briefly, I explained UBX essentially represses the formation of appendices. And it's in low concentration in T3, so there is an appendix there. It's the halt here. And T2, you have wings. There's virtually very, very little UBX, and there's no UBX in the head, so you have the antenna and the mouth structures. So that's a summary of Hox gene. I'm not going to go through this again. This is like the remaining 20% of your time. Maybe you look at this. The 80% for this chapter is that slide where you have a hierarchy. This the other 19%, 1%. Just quickly look to make sure that essentially, if you read the slide before you're done, I'm not going to gloss over, ABD, ABDB, and the concentrations. I explained that it has to do with a combination and amounts of these homeotic genes. Remember the important thing: your UBX is high, you don't have appendages, and then and then ABD, ABDB essentially further differentiate these segments. But then I went on and explained the second pair of wings in that homeotic mutant. And here's an important point, is that it's not a complete loss of function of UBX, because UBX is essential. It's an essential gene. So what these mutations are, they're mutations in enhancers that allow for the expression of UBX in both segments, constituting for parasegments. In the parasegments that constitute T3. So if you don't make UBX in the anterior end of T3, you have a wing instead of a alter, and that's for the posterior, post-bithorax mutation. And these are in regulatory regions enhancers. This is kind of like the converse experiment of the stripe 2. Right? Because I just thought of that. Just to make sure. See, this essentially we just pick just one element and we fuse it to a reporter gene. What you can do is mutate that element, and it won't be expressed in a specific part of the embryo. This is the most important. These are genes that regulate the expression of homeotic gene, and they work through chromatin structure. So trithorax and polycomb are chromatin organizers. They control histone acetylation. I'm going to stop for a second here. There are other questions regarding UBX and homeotic genes.
Can you go back to trithorax and exactly what is it? What is the use of it? Because like I was looking it up, not even in the chapter 20. I need to go back and
what's the use of these elements?
Yeah.
To express UBX in T3. And T3 is divided in two parasegments. There's an anterior half of T3, it's called parasegments, and the posterior half of T3. And there's a specific element which is an enhancer, basically. It's called bithorax, which is important for the expression of UBX in the anterior portion of T3. If you remove UBX, you're not repressing weight. UBX is a repressor. Right? But there is an appendage, though, because there's little UBX. If you were to overexpress UBX even further at T3, in principle, it wouldn't be an alter. Okay. Any other questions? So now I'm going to move to worms. I introduced that system because I thought that was not done that great in the book. So I told you that that this is hermaphrodite genetics. It's actually the power of this system is you can just with a single mutant generate a population. So I think that's not too difficult. And then I sort of alluded to that already. I talked about vulval development, and I insisted on the Lin3 pathway and on the LAC2, Lin12 pathway. The Lin3 pathway is an induction process. And Lin3 binds to its cognate receptor LET23, and that induces a signal transduction pathway in the cell that's controlled by LET60. The book, I think, forgot to tell you this is an ortholog of RAS, which is a cancer gene. It's an oncogene in humans. And in addition, this pathway leads to the expression of another molecule called LAC2. So, see, this is it here. So it's doing two things, and it inhibits transcriptionally production of the receptor of LAC2, which is Lin12. So at the same time, the Lin3 pathway and LET23 receptor and LET60, they induce the differentiation of P6P into the vulval structure, and they actually ensure the neighboring cells are induced to become supporting cells. So they will not form a vulva. And this is an inhibitory activity. Okay, so LAC2, Lin12, that's an inhibitory activity of the vulval development in P7 and in the neighboring cells as well. And that leads to the P6P expressing the ligand, LAC2 at its surface, and very little Lin12, and the secondary cells, supporting cells, P7P and P5P and etc., they express Lin12 and very little LAC2. And so you see how that development, how cells that are next to each other adopt a very different developmental program due to Lin3. That was pretty much it. Any questions? Yeah.
So before the induction happens, Lin12 present on all of those cells, right?
I have to think about that. So earlier in this developmental pathway is Lin12 expressed in these cells. You're thinking if it's not repressed in P6P, that means it's actually highly expressed or could be expressed. That's not necessarily the case. But I'll have to check on that actually, if Lin12 is significantly expressed in, for example, P6P. Before the anchor cell is making Lin3, that's what you're asking.
What about like P6P cell, but maybe any kind of cells, like any of those 6 cells?
Okay, the general answer to that question, Lin12 is important in other cell types to govern differentiation in totally different structures, and the same is true for Let60. Okay, okay, the point here is kind of what I said in the fly is also true here. You could have the same pathway, a same ligand, same receptor, generating very different outcomes in different cells because the cells, even though they respond to a ligand, are poised to induce a different expression program. And so if you're in a different part of the body, say, well, Lin3 is specific to vocal development, but Let60 will induce different responses. Okay? So I think, any questions about this slide with the double mutant analysis and epistasis tests? And the notion of gain of function, loss of function mutation, you understand that? The receptor, Let23, can have loss of function mutations or gain of function mutations with very different outcomes. If you have a loss of function mutation in Let23, the receptor, that essentially prevents either binding to the ligand or response after binding to the ligand Lin3. And so this essentially cancels the pathway, and the phenotype that results from that is vulvaless because Let60 is not activated. But that same protein, same gene, Let23, that same gene can sustain dominant mutations. And these are mutations that change the conformation of the receptor and that make the receptor adopt a conformation that normally occurs only after it has bound the ligand. So it's kind of a hyperactivated form of the receptor. And it even bypasses Lin3 altogether, and that inappropriately activates like Let60. Okay, so the same molecule, same gene, can have loss of function or gain of function mutations. The gain of function Let23 essentially is multivalent. Okay, same thing for Let50, for Let60. You can have loss or gain of function mutation. The RAS mutation in human cancers is a gain of function. And the response there is it induces cell divisions, uncontrolled cell division. That's why it's involved in cancer. Okay, that was a piece on hedgehog. Very interesting gene. So this is segment polarity gene. So that's between the perul and homeotic genes. And this was a slide that I'm not going to quiz you on. I was just trying to illustrate to you how hedgehog works and how you could have loops, feedback loops between groups of cells that allows the boundary between tissues to be made. And so hedgehogs are back to what you were saying before, it's a little bit the same thing. I mean, it's important in neural tube formation, it's important in structuring the brain, and at the same time, that molecule is very different effect or function. It allows limb development and development of the digits. And why does it do that in some cells or in one part of the world versus the other? Because these cells, they have differentiated already. They're not finished, but they are in the path where they respond differently. They have a different gene expression program with the same pathway. And I finished on that to say that there are mutations that induce polydactyly. So these are gain-of-function mutations. They're dominant. They are present in the enhancer that's in an intron, some other part of the chromosome. And that is overexpression of hedgehog. And loss of function of hedgehog also shows an apparent dominant inheritance pattern due to haploinsufficiency. Any questions about what haploinsufficiency is, or do I have to restate that? No? Restate? So, haploinsufficiency is a term to describe genes that require to be present in 2 copies for proper function. So the amount is important, okay? So a loss of function recessive mutation, if you think about it, is covered by the wild type allele that remains. So the premise here is, if we have 1 copy of the gene, that's enough for gene function. And there's no phenotype there. That way, that's how we determine that. That's how we say mutation is recessive. But some loss of function mutations are not like that. And they're not very common, but they do exist. And if you have a loss of function in one of the allele that reduces gene activity in cells such that there's not enough gene activity, it could be just because the amount of protein is not enough or the amount of activity is not enough. And then you see a phenotype even with a loss of function mutation. And you see the disease, for example, for hedgehog, these are loss of function mutations that lead to an inadequate amount of gene activity and that lead to the phenotypes that you see, such as cyclopia and in fact, incomplete formation or closure of the neural tube. You actually see essentially part of the brain coming out there too. And so an apparent dominant inheritance pattern doesn't mean you have a gain of function mutation. Okay, it could be haploinsufficiency. Is that clear? Yeah?
Yes.
Okay.
Yes.
So now we're going to make the big jump back a couple of billion years. And I'm going to just first talk about horizontal gene transfer and conjugation. So there are 2 things about conjugation: the genetic discovery and observation of conjugation, and then there's the mechanism of conjugation, and finally there's the use of conjugation as a genetic tool. Now, those are clearly the three boxes in this presentation. So the first was the Tatum and Lederberg experiment, and then you have the YouTube experiment that was done, and we can start. Maybe you can just talk about this for a second. That actually showed that horizontal gene transfer, like now we know conjugation, it requires physical contact between cells. And so if you prevent the mixing of the two strains, there's no conjugation, you do not see the formation of prototrophic colonies that are also called X-conjugates. So the only way you can see the products of conjugation is if you actually allow the cells to touch each other. And, you have to, when you look at this, look at the markers and understand the assay, okay? Because these cells are plated in what's called minimum medium. So the only cells that can grow are called prototrophic. That is, they don't have any metabolic deficiency. They can make all their amino acids and their nucleotides and their vitamins. They're bacteria, not like us. We are oxotrophic. I'm sure that's not the term, but you get the point. And so here you have two oxotrophic strains. One requires threonine and leucine and thiamine, and the other one requires methionine. So neither of them can grow in minimal medium, but if you mix them together, you create colonies that can grow because there's been gene transfer from one into the other. So, for example, the MET plus marker may have been on an F and transferred into this strain, the recipient. So you have to understand the process. And first you have to understand that what is being transferred is a circular molecule. It's called F factor. I'm asking you to read it. That's not a small detail. The size of it is 100,000 base pairs, and that's about 2% of the bacterial genome. So you need to have this order of magnitude. Remember that question on the nucleosome that I asked on the first midterm? I'm not, but I'm telling you, I asked you, what's the size? It's 11 nanometers in diameter. And, the kind of answers that I propose is the right one. And then I'll say 11 micrometers or 110 nanometers. I mean, I'm asking for orders of magnitude. I'm not going to ask you 10 or 11 or 12 or 14 nanometers. And I wouldn't do that. So same thing here. Is it 2% of the genome or 20%? You have to have this kind of order in. Another thing that, when we'll talk about the mechanism, and that's an important slide. I put in the SAM features, but there's a site called ORI-T, and that's where the nick is made by the relaxosome, and the 5' end is dragged into the recipient cell called F-. See these couple sentences that I said you have to know that. It's not the 3' end that's being transferred. It's the 5' end. And it's not a double-stranded break, it's a nick. It's a single-stranded break. A nick is a single-stranded break. And it occurs at ORI-T. you have to have in your minds also clearly the notion of F plus, which is a donor cell, F minus, a recipient cell, the notion of HFR, high frequency recombination, where the F is integrated in the chromosome. That integration occurs by homologous recombination using sequences that are present on both F and the chromosome, such as insertion sequences, as homologous sequences for that process. And then you have also the notion of F prime, which is due to an excision that uses sequences outside of the F on the chromosome. And the F can re-excise by homologous recombination, and that leads to the excision of the F with additional chromosomal sequences. So that's an extra chromosomal element containing bacterial genes called F prime. So F plus, HFR, F prime. And then I talked about the HFR matings. The transfer still occurs the same way through RBT, and the 5' end is pulled into the recipient, but this by definition will actually drag at some point bacterial sequences. And often, randomly, this will be interrupted over time, say maybe within 5, 10, or 15 or 20 minutes. The longer you wait, the higher the probability is that it's interrupted and the fragment being transferred is broken, and then that fragment actually can recombine with the bacterial chromosome, and that's how you get gene transfer and conversion by homologous recombination of phenotypes. So that's how in the Tatum and Lindbergh and the YouTube experiment, you know, if you allow cell-to-cell contact, you get production of X conjugates. And that's also a mapping technique. So that graph is important. So there are two things happening here. You see the time of appearance of the phenotypes such as azide resistance and FHT1 resistance. The time of appearance of these markers is not the same, and it depends on the distance from RET. The ones that are closer to RET on bacterial chromosome enter first. And the second thing that you see is the maximal frequency of recombination. It gets lower as you get farther from the start of transfer. And that's due to precisely the higher, the likelihood that the mating is going to be interrupted during the experiment. These are actually called controlled mating experiments, where the mating is interrupted by putting the cells in a blender. But as you allow, say, mating for 20 minutes, or 20, 30, or 40 minutes, spontaneously in these cultures, the mating gets interrupted at the moment where you plate the cells. Is that clear? Is that okay? Right. So, we discussed how this can be used for mapping technique. I just summarized that. I just explained the F prime, so you have to know what this is. And then transformation and transduction, you don't need to worry about. So this is a good 15, 20 pages that you can read this summer. Yeah. Okay. So you need to worry about this. and transduction you don't need to worry about. This is an important slide. Now that I skipped this, you need to know T4 and lambda, and you need to know these particular features. That lambda is a temperate phage that can have a lysogenic cycle, and T4 is a strictly lytic phage. So I explained that. Do you want me to explain what lytic and lysogenic is? Yep. Lytic phage, the lytic cycle is the bacteriophage infects the cells and will always, that infection will always lead to lysis of the cell. So there's immediate phage production, and that cycle lasts about 40 minutes for T4. And there is production of about 200 particles. So the bacteriophage infects the cell, and replicates its own genome, transcribes its own genes, essentially hijacks the cell, hijacks the transcription machinery, the replication machinery, and makes capsid proteins and tail proteins. There's spontaneous packaging of the DNA in the capsid. And lysozyme is also a phage gene, which lyses the cells from the inside. And someone asked me the question, and that's too much for the exam, but someone did ask me the question to discuss this is if the way the phage transcribes its genes is the same as the host, as bacteria. And the answer is yes and no. The answer is yes in the first 10 minutes of infection. So the phage promoters are bipartite and can be bound by the hollow enzyme RNA polymerase with sigma 70. So there are bacteriophage genes for T4 that actually have those promoters. But T4 has its own sigma. And so as that sigma is produced, then it will then display sigma 70 and recruit RNA polymerase to middle mode and late genes. And those promoters look completely different. So that's the answer. Lysogeny is, for example, what happens with phage lambda. So lambda, when it infects the cell, depending on the growth conditions, will face essentially what's called a genetic switch. And depending on the growth conditions on the host, there is a regulation of whether the cycle goes into lytic or lysogeny. Okay? And that if the cells are in good growth conditions, lysis, the lytic cycle occurs. And so it's exactly like T4. But if the cells don't, then the cells will integrate in the genome through an enzyme called integrase. And lambda integrase, this is not homologous recombination, it's site-mediated integration with an enzyme called integrase. And it always integrates in the same location in the genome between the genes bio-A and gal-K. And the cell can then continue to grow with the prophage, it's called a prophage in the genome, and these can be induced to resume the lytic cycle. These prophages can enter back in the lytic cycle if you irradiate the cells with UV, for example. So you don't need to worry about transduction. That I just told you about, that's integrase, it's not homologous recombination. That you need to know. Transduction with lambda, specialized transduction, you don't need to worry about that. And finally, you have the R2 locus, and that was an example of the fine genetic analysis by Sidney Brenner with using the R2 locus and doing complementation tests and doing using co-infection, and you doing deletion analysis and mutation mapping. The gist here is you can map mutations because if you co-infect with the deletion, if the mutations map outside of the deletion, then you're able to create the wild type. And this is a very easy assay because you're just asking if you have plaque formation. That's a general idea here. This is it. And this is how Brenner could recreate his map for the R2A and R2B loci. You have all these point mutations that he mapped relative to the deletions shown up here. So any questions? Yeah.
Just to make sure, are we responsible to know the molecular mechanism of phage lambda, how it decides to go into the zygenic cycle?
So that's in the other lecture. I'm going to tell you what you need to know. And why don't I just show you those slides and talk about this right now. And so this is what I want you to retain from this. The key component in the lysogenic cycle of lambda is called lambda C1. It's called C1 protein, aka lambda repressor. And lambda repressor is a transcription factor and is able to bind it to the regulatory region, complex regulatory region, which lies between its own gene, C1, shown here in green on your left, and controlling also another gene called CRO. And CRO is a lytic factor. C1 lambda repressor is a lysogenic factor, and CRO is a lytic factor. When C1 is produced, it binds to the operator shown here called OR1 and OR2, and in doing so, it represses CRO by steric hindrance, just like lac repressor represses transcription by binding to LAC-CO. Same principle. But as a lot of C1 is made, you should know that C1 is able to activate its own transcription because it acts as in OR2 as a positive factor for transcription of its own gene. That increases the amounts of C1, and C1 eventually can, when the levels are high, binds to OR3 and then represses its own transcription. And when you irradiate cells with UV activates a protease called REC-A that cleaves C1 and that liberates the operator OR1, 2, and 3, and that allows the transcription of CRO, and that leads to lysis. So what I just said is what we need to know.
Can you repeat it?
So this regulatory region with 3 operators, OR1, OR2, OR3, with C1 and CRO. So we're back to that. So we're back to the beginning of this, and we talked about the early polymerase alpha 2 beta, beta prime, omega sigma, and sigma is important for recognition of the bipartite promoter, -10 and -35 box. And there's transcription termination, which depends sometimes on row, sometimes there's row independent termination. And now I'm gonna just move quickly over, because I want to look at a couple of questions from the second midterm. Was the introduction of the helix, turn helix motifs in LAC-I, in C1, and they're also present in Hox genes. So it's a DNA binding domain that has evolved in prokaryotes. And it's a motif that has been used in eukaryotes as well. And now the lac alpha. So I think that if you remember that you have a bipartite promoter -10, -35, that's the promoter bound by RNA polymerase using sigma 70. And you need a transcription start site, right? So it's a +1. And the operator is going to be somewhere here, and that's where LAC-I can bind and prevent the association of RNA polymerase with a promoter. Then upstream that, there's the cap binding region, and this is a molecule called cap, which binds cyclic AMP, and cyclic AMP is not produced when glucose is present. So this regulation is absent in presence of glucose. And I'm going to show you an important slide. Oh, this is it. And this is why when you have glucose and lactose, LAC-I is not exerting repression, but glucose is essentially preventing the action of cap-camp, and therefore you have basal transcription. So the maximal amount of transcription is in absence of glucose, in presence of lactose. And that is, it's in absence of catabolite repression. And this is where cap is bound and activates transcription, and the repressor is not binding the operator. Yeah. How does glucose work? Glucose represses the enzyme that produces cyclic AMP from ATP. And that enzyme is called adenylyl cyclase. So glucose represses that enzyme. That's how it works. Okay? This, I did not talk about it. So you can ask me this summer. This is essentially the table I just showed you. I just put this because I think it might be a minus here in the book. I'm not sure. And somewhere that table is wrong, so it's a plus here. Now, mutations in LAC-I or LAC-O, that's an important slide. It essentially tests your knowledge. You have mutations in the DNA for the DNA sequence in LAC-O, and these, they cannot be recognized by LAC-I, and therefore LAC-O mutations are called constitutive mutations. The promoter is always on. It cannot be repressed by LAC-I because its cognate sequence is mutated. So these are called OC, or O constitutive. You can have also just loss of function mutation in like repressor, such that it cannot bind to LAC-O, and these are also constitutive because you don't have, LAC-I is not exerting the repression as it usually is. So this is, in absence of glucose, you still see the operon is still on, right, when it should be off in wild type. You have mutations, and these are called super repressor, and these are mutations that lead to the overexpression of LAC-I, and therefore it is bound, if you will, in a higher at a higher frequency to LAC-O, and these are usually mutations that lead to uninducible phenotypes. And there's also here a description of a mutation that prevents binding of LAC-I to inducer, and the same thing, it'll be bound to LAC-O, and therefore this would be uninducible. Essentially, LAC-I cannot respond to inducer, and is always bound to the operator. That was an illustration for illustration purposes. That's also a little bit more than you need to know. That it's an inverted repeat. So helix turn helix, that's important to remember. That's the same figure I showed you, but in a slightly different design. I will not ask you anything about the trip opera. And I just talked about this. Questions? Yeah.
Sorry, can you clarify again
The significance of the helix turn helix motif is that it's a recognizable common DNA binding motif that has appeared already in prokaryotes, in LAC-I, for example, and that's a mode of DNA binding that can be recognized based on the amino acid sequence, and that is also found in eukaryotes. So now I'm gonna go through a couple of exam questions to sort of refresh a bit of memory for the second part. And I'm going to just solve with you a couple of these things and see, to sort of put you back in the mode of this test. So this is a question that was in the second midterm. You cross a female with white eyes and a male with red eyes, and you notice a very low frequency of male progeny with red eyes. So that's funny, because if you have a female with white eyes, all the males should have white eyes in principle, right? So that's kind of strange. It's a very low frequency event. So how can that happen? So this was a slide that was taken from the book when the book explained about non-disjunction. So I was asking you to, so that's the answer. The term is non-disjunction, or chromosome non-disjunction, or non-disjunction in meiosis 1. Fill out the Punnett Square indicating the female gametes, given they give you the normal male gametes here, X, Y, and it's Y plus, so this is a male with red eyes, and expected 4 genotypes corresponding to this rare occurrence, assuming it occurs during meiosis 1, so I give you that. So if you have non-disjunction, the female gametes are going to be either XX, so that's the non-disjunct X chromosome containing the Y gene, and the other gamete, when this happens in meiosis 1, will not have a sex chromosome. So I indicate that by 0. So then how do you fill out the Punnett square on top here? So if you have the non-disjunctive X chromosome, you have XXX with, if the sperm has an X chromosome, and XXY if the sperm has a Y chromosome. Then at the bottom, without X chromosome, you have X0 and Y0. And then I told you that implies X0 is a male, so this is a male, and XXX and Y0 are lethal. So this is lethal, and this is lethal. So now if you know this is male, you'll notice that this has the Y plus chromosome, right? And that's how you get this very unusual outcome of male progeny with red eyes. That's how it happens. And the funny thing, if I can call that funny, is that the male inherited X from his father. Whereas like this, normally never happens that X is inherited maternally in a normal male fly. But in that case, you get a male outcome, and the X comes from the father. And that X has the wild type Y gene. And then the other one that's viable is XXY. And this is homozygous, in fact, white minus X. So this is a fly that has white eyes. And the answer to the other rare progeny that you expect is a female with white eyes, because in flies, XXY is female. But those of you who did the Punnett Square correctly and found that this was the other genotype, but you thought this was male, I did not hold this against you. But in humans, it's male. XXY is male, but in flies, it's actually a different way to determine the sex, so it's female. So if you told me male with white eyes, I said that's okay. I didn't deduce anything. But the real point here is to understand how that non-disjunction occurs in meiosis one. What are the gametes that are formed? Okay. Now, here's another question. That's a question where I was asking you, if you have a cross, like how many out of 1,000 flies, how many recombinant flies do you expect? And then I think that in some cases the issue is a bit of a vocabulary issue. So let me read the question. The recessive mutations, hairy wing, and purple eyes are 8 map units apart from chromosome 2. Phenotypically wild-type females, HW plus PL minus over HW minus PL plus, so they're phenotypically wild-type, but they're heterozygous mutants, were mated to hairy wing males with purple eyes, so they're homozygous mutant, HW minus PL minus, homozygous. Which of the following phenotypic crosses reflect offspring that were generated as a result of a crossover event? So, the male is irrelevant, okay? The male is homozygous. So the crossover is not going to affect anything. Plus, in flies, for whatever reason, there's no recombination in meiosis. So you consider only the female in this case. And so this is situation female, right? So this is the genotype, HW plus PL minus and HW minus PL plus. This is meiosis 1, so we have duplicated chromosomes. And let's consider recombination between 2. So this is how you get recombinant for these markers, is when you have recombination between the 2 markers. So the recombinant chromatids, so we have chromatids 2 and 3 here, recombining, are going to be HW, and now follow my laser as you can. It's going to be HW minus, PL minus, And the other chromatid recombinant chromatid is going to be HW plus, PL plus. These are the recombinant chromatids. So what happens when there's recombination? You're producing recombinant types by mating with that male. The male contributes a gamete that's HW minus, PL minus. So the recombinant types are going to be the male double mutant with this one, HW plus, PL plus. That's the wild type for both the phenotypes. So normal wings, normal eyes. And then the other recombinant in the female is going to be the double mutant with the double mutant male. That's the double mutant phenotype, hairy wings and purple eyes. And so the recombinant, the phenotypic classes reflecting offspring generated as a result of a crossover event are going to be the double mutant and the wild type. So the answer is B and C. Okay, so far? So now I'm asking, if you have 1,000 flies in the offspring, in what number would you expect the following phenotypes? And these are the same phenotypes, and so you know these. You know already that this is the recombinant and this is the recombinant because you've already saw that for question 1. And the single phenotypes are the non-recombinant or parental types. Parental because these comes from unrecombined chromatids in the female, the mother. So you know they are 8 centimorgan or 8 month unit. So that means that the number of recombinant types which you've already defined in the cross is going to be 8%. Out of 1,000 flies, 8% is 80. You have 2 recombinant types, so you're going to have 40 of one and 40 of the other. So you expect 40 wild type and 40 double mutants. The rest is going to be 920 non-recombinant types, and that's 460, 920 divided by 2 for each type. So you're going to have 460 for hairy wings and 460 for purple eyes. Okay? I'll do one more. Let me just go through these questions, these 2-point questions, because I have the feeling that, you know, several of you told me, you know, you understand it, and you understand the answer, but then you look at your exam, you're like, I can't believe I answered that. And a lot of the time, it's really a technique of answering these questions. And I know it's nerve-wracking, but I want to say there's always one clear answer. And I know it's easy to say, and I know it's not easy to take this exam, but we're not trying to trick you. We're trying to test your knowledge, and we're asking you a lot of these questions, but these questions are not trick questions. They're not like twisted or things that you need to think of all the possibilities. Okay. So for example, in meiosis, reduction of ploidy occurs. I told you it's a meiosis 1 issue. That's where the homologs separate. That's textoid, one of the slides. Which feature is not relevant to balancer chromosomes? So, dominant markers that's relevant. I talked about the bar mutation, recessive lethality as well. Paracentric inversion is relevant. The P element is not relevant. P element is a transposable element. Again, there's a clear answer that's outside of the other 3. Another P element question. P element Is active in all cells? No, it's active only in pole cells, so you cross that out. Is a functional transposon carrying transposase? Maybe. Is active crossing of P female? Cross that out, has to be an M female. Is functional carrying the reverse transcriptase? No, it's not the right enzyme, so the answer is B. So I think that when you, perhaps it's helpful when you read these questions, something looks false, just cross it out. And if you end up having one question that makes sense to you, that's probably the right answer. There was someone asked me about tetrad analysis. You can come see me tomorrow if you want to review that. This is, so this I know is a difficult question. It's not tricky, but you need to read it carefully. Read your exams carefully. Read the question very carefully. So it's important. For example, tetrad analysis, which statement below is true? If two genes are linked, that's the key thing, okay? So here's one. The number of PDs and NPDs is exactly the same across this. This only occurs when they're unlinked in different chromosomes. Tetrad types result from recombination between centromeres and the marker. That's irrelevant when the genes are linked. Parental ditypes cannot occur through recombination. That's not correct. I know that's a little bit technical, but why would parental ditypes not occur? NPDs are the least abundant category. That's because they require 4-strand double crossovers. So that's actually technical. I know you have to remember this. Again, there is a clear answer. There's no question. But anyway, I wish you the best of luck for Tuesday. I'll see you at 5:20.
Do you want a copy of this lecture?
Copy of what?
I recorded the lecture.
Oh, really?
Yeah.
Wow. Cool.
Do you want it?
Yeah. Good. Thank you.